Welcome to Section 1 of Genetics. This section will provide an overview of many basic genetic principles. Let's get started. This is a table of many basic genetic terms which can be found in Section 1 of Genetics. This table is by no means a list of all the terms you need to know, but it's a great starting point and includes some of the highest yield terms to be familiar with for Step 1. Throughout this video, we'll go through each term and I'll provide examples to help solidify the information. Okay, let's start with codominance. Codominance refers to the idea that multiple alleles associated with a gene are simultaneously expressed. Blood groups are a classic example of codominance. Let's draw this out so you can see what I mean. First, I'll draw 4 red blood cells. Next, we'll add the antigens. Okay, notice that patients who have the A gene or have type A blood only express the A antigen on the red blood cell surface. So these green triangles represent the A antigen. Individuals with the type B gene only express the B antigen. So the purple square represents the B antigen. However, type AB express both the A and B antigens. This idea of simultaneously expressing multiple alleles from one gene is called codominance. So this example represents codominance. I've also shown off to the right type O blood. Patients with this type of blood don't express the A or B antigen. Incomplete penetrance is defined as individuals who carry the mutant genotype but do not express the mutant phenotype. For example, one patient with the BRCA1 mutation may develop breast cancer, while the other patient will remain cancer-free for her entire life. So again, this idea of some patients carrying the genetic mutation but not actually manifesting the disease is called incomplete penetrance. Variable expressivity refers to the idea that there is variability in the phenotype expressed by individuals with the same genotype. For example, some patients with neurofibromatosis type 1 may have cutaneous neurofibromas covering the entire body, while others may only have a small part of the body covered by lesions. Don't get this confused with incomplete penetrance. These patients will all have some type of manifestation of the disease. It will just differ in severity. So notice that these individuals all have 100% penetrance. This is an image of neurofibromas. Notice that this patient's back is completely covered in these lesions. This is a classic image of neurofibromatosis type 1. Pleiotropy refers to the idea that one gene is responsible for multiple phenotypic manifestations. For example, in homocystinuria, patients present with many symptoms affecting multiple organs. Some findings include lens dislocation, DVTs, a marfanoid habitus, and intellectual disability. Anticipation is defined as the tendency of a disease to occur earlier or to be more severe in succeeding generations. A classic example of this is Huntington disease. Huntington disease is a trinucleotide repeat disorder caused by CAG repeats that accumulate in DNA. Symptoms of Huntington disease include dementia, chorea, and behavioral changes such as aggression or depression. The disease can cause anticipation because the CAG repeats are increased during spermatogenesis. Therefore, if an individual inherits this mutated gene, then they will have more CAG repeats compared to their parent. This process continues throughout generations and the CAG repeat grows, resulting in symptoms that are more severe or that occur earlier in succeeding generations. So again, this concept is called anticipation. We'll talk more about Huntington disease in a separate section. Loss of heterozygosity refers to the idea that tumor suppressor genes must develop mutations in both alleles before cancer will develop. A classic example of this is retinoblastoma. Let's draw this out. Normally, we have one maternal chromosome 13 and one paternal chromosome 13. Each chromosome contains a tumor suppressor gene. If a mutation occurs in one chromosome, the opposite chromosome still functions and suppresses tumor growth. However, if a mutation also occurs in the other tumor suppressor gene, then both genes are disrupted and tumor growth is no longer suppressed. So, retinoblastoma is a genetic disorder that results in pediatric retinal tumors. Patients with retinoblastoma are born with one inherited mutation of the tumor suppressor gene in all of their cells. It's normally very unlikely for one cell to have 2 somatic mutations in both tumor suppressor genes. However, in these patients, only one somatic mutation is necessary for tumor growth to occur because they inherited a defective version of the gene. This is a classic image of a patient with retinoblastoma. Notice that the patient's right eye has the normal red reflex, whereas the red reflex is absent in the left eye. You can see the white reflection right here, which represents growth of the tumor. Mosaicism is defined as the presence of multiple genetically distinct cell lines in the same person. This can be further divided into germline and somatic mosaicism. Let's look at some figures to understand this better. This is a figure illustrating germline mosaicism, which can be found in section 1 of genetics. Notice from the image that this occurs when a mutation occurs early on in a progenitor gamete cell. In this figure, a progenitor sperm cell is shown, but this could also occur in a progenitor oocyte cell. Because the mutation occurs early on in the development of gametes, it continues in many generations of daughter cells. Ultimately, this results in an individual with 2 distinct cell lines in their gametes. In this particular illustration, we've shown half of the gametes containing the mutation and half not containing the mutation. Depending on how early the mutation occurs will determine how many daughter cells are ultimately affected. In germline mosaicism, the individual will not manifest the disease, but his or her offspring will manifest the disease if a mutated gamete is inherited. So from the figure, you can see that we've shown a man whose gametes appear abnormal, but the other parts of his body are normal. Notice that his offspring can be normal or can inherit the mutation depending on which sperm combines with the oocyte. This is a figure illustrating somatic mosaicism, which can be found in section 1 of genetics. Notice that this occurs when a mutation occurs early on during embryonic development. For example, if a mutation occurs when the morula is dividing into the blastocyst, this can result in a mixture of abnormal cells and normal cells. In somatic mosaicism, the individual may manifest the disease, but his or her offspring will not manifest the disease because the gametes are unaffected. So from the figure, you can see that we've shown a man whose gametes appear normal. However, the rest of his body is abnormal. Also notice that neither of his offspring are affected. Finally, you should know that sometimes the germline and somatic cells can both be affected, resulting in combined mosaicism. In this case, the parent is affected and his or her offspring can also be affected. So it's kind of like a combination of the 2 images we've just shown. From the table, notice that Turner syndrome and McCune-Albright syndrome are associated with somatic mosaicism. These are 2 commonly tested syndromes associated with mosaicism, but many other disorders are possible. For example, sometimes trisomy 21 or Down syndrome can be caused by somatic mosaicism. Turner syndrome is a sex chromosome disorder that affects females. These patients only have 1 X chromosome, so 45XO. McCune-Albright syndrome is caused by a defect in G protein signaling that occurs after fertilization. In other words, it's a form of somatic mosaicism. Findings include café au lait spots, precocious puberty, bone abnormalities, and endocrine organ dysfunction. Okay, let's do a question so you can see how this will show up on exams. A 1-day-old boy is born to a healthy 25-year-old female at term. On exam, the boy is noted to have upslanting palpebral fissures, a single palmar crease bilaterally, and prominent epicanthal folds. FISH analysis on cultured fibroblasts shows that 70% of the cells have 47 chromosomes, while 30% of the cells have 46 chromosomes. What genetic principle most likely explains these findings? A. Meiotic non-disjunction. B. Germline mosaicism. C. Somatic mosaicism. D. Loss of heterozygosity. Or E. Variable expressivity. Hopefully from the question stem, you notice that this boy has signs and symptoms consistent with Down syndrome, or trisomy 21. These include upslanting palpebral fissures, a single palmar crease bilaterally, and prominent epicanthal folds. Because FISH analysis reveals some cells with 47 chromosomes and others with 46 chromosomes, we can conclude that there must be 2 distinct genetic cell lines. As we've been discussing, this is also known as mosaicism. In this example, somatic mosaicism can explain these findings. So the answer is C. Meiotic non-disjunction is the most common cause of Down syndrome, but this would result in 100% of the cells containing 47 chromosomes. If the boy's parents had mutated gametes due to germline mosaicism, this could have caused Down syndrome, but again, 100% of the boy's cells would have contained 47 chromosomes. Loss of heterozygosity refers to the 2-hit hypothesis of cancer, and variable expressivity refers to the idea that there is variability in the phenotype expressed by individuals with the same genotype. So again, the answer is C. Okay, let's continue discussing the table. Locus heterogeneity refers to the idea that the same phenotype can be caused by mutations at different loci. An example of this is albinism. Alleic heterogeneity refers to the idea that the same phenotype can be caused by mutations in the same locus. A great example of this is beta thalassemia. Let's draw this out. This will represent 1 chromosome we'll call chromosome A, and this will represent another chromosome which we'll call chromosome B. Now let's say that on chromosome A, a mutation occurs right here. On chromosome B, a mutation occurs right here. In locus heterogeneity, either of these mutations will result in the same phenotype. In alleic heterogeneity, mutations affecting different parts of the allele can result in the same phenotype. So for example, let's assume that this represents an allele. In this example, mutations to the promoter region, the introns, or other parts of the allele could all result in the same phenotype. So for example, a mutation right here, or right here, or right here could all result in the same phenotype. Again, this idea is called allelic heterogeneity. Okay, moving on. Heteroplasmy is defined as the presence of both mutated and normal mitochondrial genomes within a single cell. Mitochondrial disorders are classically associated with heteroplasmy. This is a figure that illustrates heteroplasmy, which can be found in section 1 of genetics. Notice that there are both normal mitochondria and mutant mitochondria shown in the image. In some patients, there will be very little or no mutant mitochondria, resulting in unaffected offspring. On the other hand, some patients will have many mutant mitochondria, resulting in severely affected offspring. Okay, the last term to be familiar with is dominant negative mutation. This refers to the idea that a mutation can result in a dominant effect. So for example, a mutation could result in a non-functional protein. If this mutated protein prevents another normal protein from functioning properly, then this is said to be a dominant negative mutation. Okay, let's do a question. A 41-year-old female presents to the physician for chronic muscle weakness. A skeletal muscle biopsy reveals ragged appearing muscle fibers. DNA analysis reveals a mutation in 70% of the mitochondrial DNA. The physician is concerned about the possibility of the disease affecting the patient's children. However, the patient states that none of her children have any symptoms. What genetic principle most likely explains the absence of symptoms in this patient's children? From the question stem, we learn that this patient has chronic muscle weakness and a mutation in 70% of the mitochondrial DNA, but none of her children have any symptoms. Mitochondrial diseases are passed on maternally, so we'd expect some of her children to have the disease. However, heteroplasmy can explain this discrepancy. Because 70% of the mother's mitochondrial DNA contains a mutation, 30% must be normal. This 30% can be passed on to her offspring, resulting in unaffected children. So the answer to the question is heteroplasmy. You don't need to know very many specifics of mitochondrial diseases, but you should definitely know that they result in muscle damage. And this should make sense, right? Skeletal muscle tissue has a lot of mitochondria, so skeletal muscle tissue is usually affected in patients who have a mitochondrial disease. It's also worthwhile knowing that a muscle biopsy will usually reveal ragged appearing muscle fibers regardless of the type of mitochondrial disease. So again, the answer to the question is heteroplasmy. Again, this is a figure that illustrates heteroplasmy. Notice that there are both normal mitochondria and mutant mitochondria shown in the image. Although this patient was affected, her offspring were not. In this case, they must have had a lot of normal mitochondria. This is a figure of a Gamori trichrome stain that shows ragged appearing muscle fibers. This can be found in section 1 of genetics. You can see the arrows right here and right here pointing to the ragged fibers.